Hello, everyone. I hope you all are doing well. Miss Hafsa, am I audible?
Yes, sir, you are audible.
Okay, all right. Miss Jannatul Ferdous, my voice is clear to you? Miss Jannat, can you listen me?
Yeah, I can listen you, but the sound is very, small. Or is it my problem or yours?
Yeah, maybe my problem. Here is Hafsa today hosting this class. So, Miss Hafsa, right now you can start, please. And yeah.
Okay,
Welcome
sure, sir.
all of you in today's class. Best regards.
Assalamu alaikum, everyone. Hope so I am audible to all of you. And I hope you all are having a wonderful and relaxing weekend. Thank you for joining us today despite your busy schedules. It's a pleasure to welcome you all to the fourth class of Advancements in Molecular Biology course 909. We are delighted to have such enthusiastic participants with us today. Today, we are honored to have Ms. Jannatul Ferdous, our distinguished biotechnology lecturer from the University of Science and Technology, Chittagong, Bangladesh, who will be sharing her expertise and insights with us. And you guys, if having queries, you can write in the chat. We will sure acknowledge them at the end of the session. So let's move towards the session. Now, I would like to hand over the class to Ms. Jannatul Ferdous. Ma'am, over to you. You can take start now.
You have some. The slides
Hello,
are
ma'am.
not showing. Hafsa, slides are not showing clearly.
Actually, ma'am, it's clear from my side. Let me check again.
Okay.
Is right now clear?
Okay, let's go. Yeah.
You can move forward.
Okay. So welcome to all of you, and thanks Vita Nova, for this session. So I can share some of my learnings with you guys. So as Hafsa said, I will share my expertise. Sorry, but in that case, I am not expert. So I will try to share what I have learned about this specific technology in molecular genetics. What which is doing so much favor in our life without our knowing. There are many molecules in our body that fight even we don't know. Some sort of like lysozyme. We we don't know how it works or when it works, but as we learn science, we can know that they are protecting us every day from various microorganisms. So today we are going to learn about one of them, which is as much as like important like CRISPR, so much hype with the CRISPR, but I think siRNA is also a hero in our life that is protecting us in every way. So, as I said, that today's topic will be on siRNA or small interfering RNA. We'll learn how it works, what is the mechanism, how siRNA can help us in our molecular field. Then we will learn who works on it, how it won Nobel Prize, and then its therapeutic uses. Can we use it at therapeutics, or what hurdles we may overcome or we may face in this process? I will try to explain a little bit. So I will hope everyone will learn something from this class. Before going to the main session, I would like to introduce myself. I am Jannatul Ferdous. I completed my bachelor and master's in genetic engineering and biotechnology. Now I am working as a lecturer in Department of Biochemistry and Biotechnology. Actually not just biotechnology. My department name is Biochemistry and Biotechnology. So that's it, nothing more. So let's move on. Next slide. Hafsa, next slide. Next slide, please. Okay,
Actually, I
thank
have
you.
both the slides.
Okay. So the process by which a siRNA or a small interfering RNA works, known as RNAi or RNA interference. This is actually an endogenous process where the small RNA molecules, which the size is usually based from 20 or 21 to 30, not more than that. So RNA molecules within this region can silence gene expression of a target gene. So they target a specific mRNA and then neutralize it. So when the mRNA is neutralized, the protein cannot be synthesized. Thus, it can be used in gene expression regulation in synthetic process. If we want to, downregulate or inhibit the expression of any specific gene we want, then we can use this process. Also, it is working as a defensive mechanism against viruses, as we know virus contain RNA. So the RNAs can also act as siRNA in this RNA pathway to protect us from viral infection. So actually there are three types of RNAs. Fourth slide. Hafsa, fourth slide. Yes. So there are actually three types of RNAs that work in this pathway. First one is siRNA or small interfering RNA, then miRNA or microRNA, and another one is piRNA. All of them are a small RNA molecule. They are non-coding RNA molecule, work in the same pathway, RNAi pathway, but their working mechanism and their size differ. That's why we have three different types of RNA. All of them downregulate the gene expression of any specific gene. So today we'll look at siRNA only because after the session we will talk about why it is more important than other RNA. So in case of a small interfering RNA, the size is known to be 21 nucleotide. In this 21 nucleotide, 19 nucleotide are, base paired, but there are 2 nucleotide overhangs in the 3' region. And siRNA can be synthesized chemically or enzymatically. They are double-stranded RNA. They are complementary to the target mRNA. And as they are complementary to target, as we said, they can knock down any specific gene, and they can also preserve the genome integrity. If we are attacked by any virus or any bacteria, and the genome of the microorganism is trying to enter our genome, then siRNA can help in this process by degrading the mRNA of that microorganism's genome. So indeed, in this case, they can help us in preserving our genome integrity. So before we know how siRNA works, we will look into some proteins and complexes that are vital for this mechanism. One of them is dicer protein. Dicer is known as molecular scissor. You all know what is molecular scissor is. We know about restriction enzymes. we know about catalytic antibodies. All of them are molecular scissors. So what molecular scissor does is that they cleave any specific DNA sequence or RNA sequence. Dicer does the same work. It has several domains. In some domains, it binds to the double-stranded RNA, and some domains like RNA's 3-domain cut the double-stranded RNA. And then here comes the Argonaute protein. It is said that Argonaute is the, central player. Dicer cuts, but the main role played by Argonaute. It is the central player of this process. So it is also a RNA. It is all like a protein that is known as silencing effector, and the guide RNA, we will see where the guide RNA we find. The guide RNA or small RNA will guide it to the target mRNA. So Argonaute also have multiple domain. The double-stranded RNA, their siRNA that is produced by the dicer will bind to the domains of Argonaute, and the process will continue by the Argonaute. There is a complex which is known as RNA-induced silencing complex. This complex helps Argonaute to continue the further process, which means you got your siRNA from the double-stranded RNA by the help of dicer. Then Argonaute comes and binds the siRNA. The risk bind and Argonaute helps you to, cut the mRNA. So there are actually the 2 protein and 1 complex. Dicer, which acts as molecular scissor, then Argonaute, which is the central player, and there is risk or RNA-induced silencing complex, which contain Argonaute and other protein, helps the mRNA to degrade, ultimately silencing it or inhibiting the protein production. Next. Okay. So I will request everyone to look at the figure on the right side. On the very right side, you can see there are a double-stranded RNA or a hairpin RNA. There are 2 RNAs. So at first, what will dicer do? Dicer will come and recognize the double-stranded RNA, bind it, and cut it to produce a 21 nucleotide short RNA. So you had a long RNA, dicer came and it cut the RNA in 21 nucleotide RNA. So we said siRNA is 21 nucleotide. We named this siRNA, a small interpairing RNA. Then when we get the siRNA duplex, here comes Argonaute. What Argonaute will do? Argonaute will bind the double-stranded siRNA duplex and it will select only one strand. As we have 2 strands, Argonaute will help us to select only one strand and it will remove the other strand. So the strand that is selected and bound by the Argonaute is known as guide strand, and the strand that is removed is known as passenger strand. So from the duplex, where is one is guide strand and other is passenger strand, passenger is gone, guide is bound to the Argonaute. We at the very first I said that the RNA will be complementary to the mRNA. So here the guide strand must be complementary to the mRNA. We want to downregulate or down its expression. So here the guide strand will bind to the complementary mRNA with the help of risk. What risk is? Risk is Argonaute protein and other proteins that house the binding of the guide strand to the target mRNA. So now I have risk, Argonaute, and guide RNA plus the target mRNA. Argonaut have the same RNase activity by which it removed the passenger strand. Now it will cut the target mRNA. So the mRNA is clipped. It cannot translate further and no proteins in the consequence. So the gene is silenced. So from the double-stranded RNA, we got the siRNA by the help of dicer. Then it formed a guide RNA and Argonaut complex that bound to the target mRNA, and then it cleaved the mRNA, thus gene silencing. So it is the main or whole RNA pathway in which the siRNA works. mRNA and piRNAs also does gene silencing, but there are some differences in their pathway. Like mRNA not only clip the mRNA, it can also, downregulate the post-transcriptional mRNA in a low level, not wholly downregulate it. It can allow very small amount of translation, whereas siRNA fully downregulate or knock down the gene. So there are some differences in their mechanism or in their way of acting. That's why there are different types, and their size varies. Like siRNA is more 24 or 25 nucleotide. Sorry, miRNA. Whereas siRNA is only 21 nucleotide. siRNA specifically work on one gene at a time, but mRNA can work on multiple genes at a time. So there are multiple differences among them. Next. So from this slide, I tried to show the mechanism in more detail. Same mechanism, same process, just, as at first I said, we will have a double-stranded RNA in the cytoplasm, which is long. Then dicer come, it shows this RNase activity and clip the long double-stranded RNA in short double-stranded RNA. In short double-stranded RNA, there are two nucleotide overhangs in the 3' region, and with the total size can be 21 to 25. In siRNA, I am saying again, it can be 21. Some article says it can be 25, but most of the time it is 21. In most articles, it is like 21, mentioned 21. Not very, yeah, 21 is the most prevalent, and some said it can be 25. So in the next slide, we can see that the guide RNA and passenger RNA duplex bind to the dicer and TRBP complex. Then here Argonaut comes. What Argonaut does, I said Argonaut will clip the passenger strand and remove it. It will bind to the guide strand, and here we mentioned TRBP. TRBP is a protein of risk. This is not a single protein. I said it is a complex. So beside Argonaut, there are several other proteins in the risk. TRBP is one of them. In the next slide, we can see that guide strand bind to the target mRNA, Argonaut cleave it, and the mRNA is degraded by the exonuclease activity. So there is no protein, no phenotype of the gene is silenced completely. This is the mechanism of siRNA in RNA pathway. So if you ask me where we get the siRNA, what are the sources of siRNA? So siRNA can be found in our body, or we can synthesize it in the laboratory. So as we can see here that like transposons, viral RNAs, bidirectional transcription, if any gene, if bidirectional transcription occur in a normal gene, it can produce a double-stranded RNA. Dicer can recognize it and produce siRNA. So different types of double-stranded RNA can be found in our cytoplasm or in our body. dicer can recognize them and produce short siRNAs. And the mechanism is same exactly as I said. So there is nothing new. Next, move on the next slide. Hafsa, next slide, please. Hafsa.
Yep, slides are moved.
Oh, okay. Should I continue without?
Yes, please continue. I am not getting the point why it's not visible to you.
Okay, no, it will be great if we can see the slide because I need to point out some points. That's why.
Here, yeah, mechanism of siRNA mediated gene silencing. Yeah, that slide showing on.
Next, like 13th number. Next. Yeah, this one. In this slide, I tried to visualize the discovery and the work done by siRNA technology and the work that won the Nobel Prize. So at first, at very first, the siRNA was detected in a flowering plant that was a transgenic plant. But at that time, scientists didn't know it was actually a RNA pathway. Um, they tried to produce a deep purple flower, but when they mixed the gene responsible for the deep purple color more, they observed that instead of giving a deep purple flowers, the plants produced white, completely white, or strip of white and purple colored flowers. So it was very surprising. So they named it as co-suppression. Like they thought that one gene suppressed the other gene, something like that. The siRNA pathway was not clear at that time. After that, in 1998, Fire and Mello did an experiment on C. elegans where they first coined the name RNAi pathway or RNAi technology. So from that, from then it was clear to the scientists that there is not any gene involved. Actually, a RNA is involved. A small RNA is silencing a specific gene. From then 1998, siRNA was found in the plant, and 2001, first synthetic siRNA was produced, and it was injected in human cell. From all the study, they concluded that not a single stranded RNA, but a double stranded RNA is responsible for the gene expression silencing. So among all of them, Fire and Mello won the Nobel Prize because their experiment pointed out clearly that double stranded RNA cause gene silencing and not any co-suppression or any other process, but it is a RNA mediated silencing process. So for their discovery, they were rewarded with the Nobel Prize in 2006. So what the Nobel laureates did, what was their experiment? Andrew Fire and Kerrig Mello. So they did experiment in C. elegans. We all know it is a model organism, very transparent, can be seen easily, any modification. So, and the genome is very, small and not very complex. So in that case, they injected single stranded RNA, double stranded RNA of a specific gene. Like here we can see MAX3 gene. So the siRNA of MAX3 gene was injected in two form. One is single stranded form and other is double stranded form. They observed that when in the C. elegans that were injected with single stranded RNA showed no effect, but the C. elegans with double stranded RNA injected showed the effect. Like so, from the MAX3 siRNA experiment, they showed that mRNA is disappeared. It explained that actually what happens to the RNA. So is it DNA is degraded or gene is removed? What happens? So they saw that transcription occurs of the MAX3 gene, but the translation is not. So and the injected double-stranded RNA bind to the target mRNA, not to the DNA sequence, but the mRNA sequence, and the mRNA is removed by the exonuclease activity. So this was the experiment that showed that the regulation happened in the mRNA level. In the same time, they did also another experiment where they worked on another gene known as UNC22. UNC22 is responsible for muscle contraction in the C. elegans. So if we silence the UNC22 gene, the muscle will not be contracted, but as it does in normal C. elegans. So similar case, it showed that single stranded RNA cannot produce any effect, but double stranded RNAs can produce. UNC22 gene showed what happens like to the animal, like what happens to the C. elegans if we silence a gene. And the MAX3 experiment showed in which level the actually regulation occurs. So at the same time, they did two experiments. And from the experiment, they proved, as I said, that RNA actually responsible for gene regulation, and there is no co-suppression, actually a RNA-mediated gene silencing process. For this work, they were given the Nobel Prize. Next. In this slide, you can see the history. I tried to show the history from the 1998 to 2024. It was the latest I got in the internet. So in 1998, discovery of RNA in C. elegans. Then 2006, it was Nobel Prize. Then 2010, you can see clinical trial failures. Then there are different types of ups and downs in this process, in this like 25 years or 26 years journey. But the groundbreaking discovery was when FDA approved first siRNA drug in 2018. In 2018, FDA approved a drug for human use. Then in 2019, there was a conjugated siRNA drug approved. So 2018 was the breakthrough, and after that, many drugs were made available in the market for human use. So scientists now are targeting different organs like liver, brain, kidney, heart to treat different types of neurodegenerative disease or like genetic disorder by siRNA technology. So why we need siRNA? Like, we can use different types of medicine. If this medicine doesn't work, we can use another one. So why we need siRNA? So they are saying like there are many drugs that cannot be reached in brain, different parts of our brain, because of their size or because they don't have so much half life to reach there. So different types of limitations are in our regular medicine. So, and there are also some side effects of medicine. So as we said, siRNA can silence a gene specifically. So if we can inject siRNA to silence a specific gene in the brain, then it can, prevent the other offsite silencing or offsite targeting of drugs. It will work on this specific gene only. But in that case, the design have to be precise. It is not that easy as we say. In that case, the Alzheimer disease will be, cured by now. So it is not that easy because there are several hurdles we need to face. Some of them are like half life, and then there are some nucleus that can degrade the RNA in our body, in the cytoplasm, in the serum. So there are many challenges we have to face when we think about using siRNA as a medicine or drug. 17th slide. Hafsa 17th. Next, yeah, this one. Here you can say I mentioned around 5, barriers like serum nucleases. As I said, in our blood there are some RNA nucleus that can rapidly degrade the RNA. Then there are RNA is negatively charged. Also, our cell membrane is negatively charged. So the negative repulsion occurs, and siRNA cannot be uptaken by the cell. Also, in some cases, body can target it as foreign molecule and entrap it, like phagocytosis or other process can destroy it. Also, some cases siRNA can stimulate the innate immunity. So there are very precision required to use it as a drug molecule. So what can we do? If we look at the barriers only, it is impossible to, use siRNA as a drug, because the drug we use nowadays also have limitations, but we did some modification in them. That's why we are using them. So scientists followed some ways or pathways to modify the siRNA. Some of them are like chemical modification. In the next slide, we can see chemical modification of siRNA. We know RNA is composed of a sugar molecule, ribonucleic acid. So if we modify the ribose sugar, it can produce a chemically stable siRNA. So what will happen if we modify them? It will increase, first of all, it will increase the half-life. Next slide, Hafsa. Yeah, thank you. So here we can see some chemical modification like 2' oxygen methylation or backbone modification, unlocked nucleic acid modification. Different types of modification will produce a very stable and clinically viable siRNA that can be used as therapeutics. So ultimate result of this modification will happen that it will prolong the half-life. We are very concerned about the half-life of our drugs. As I said, there are different types of drugs that cannot be reached to the different parts of our brain because the potency of it is destroyed before it reaches the target site. So the modification can increase the half-life of siRNA. Then also, if we make the siRNA positive by chemical modification, the negative phospholipid bilayer will not repulse it. It will uptake the siRNA, the positive siRNA. Then also the chemical modification help to resist the degradation by different types of nucleases in our body. Next. Next, skip this one. Go to the next one. Yeah, thank you. So one is chemical modification. Then we can use like liposome to deliver the siRNA lipid. So liposome is like multiple layer of lipid. We will coat the siRNA with lipid layer. So in that case, lipid encapsulation will help the siRNA to enter into the cell. The cell is also lipid bilayer, so the lipid will easily enter into the cell without any contradiction. And so it will protect the liposome barrier will also protect from enzymes activity that will degrade the siRNA. So this is another way to deliver the siRNA. Third one is like we can use nanoparticle. We are using nanoparticle in various sectors in our science. So nanoparticles can also be used in this case. Lipid nanoparticles can be used. Synthetic polymers. Here is one example like PLG encapsulation. So the encapsulated siRNA are protected from the surroundings. It can not be degraded by any nucleus, or the cell will not repulse it. So all of these modifications help to reach the siRNA in their target site in a given time, overcoming the hurdles that we face in case of available drugs. Also, it also has the special, privilege that it will not show any side effect. In most cases, will not show any side effects, as it will only regulate one specific gene that we target. Next. Okay. So what are scientists doing with this technology? They are trying continuously. The most cases, in the most research, they are actually targeting on liver or hepatic cells. Like you can see here, current clinical targets are PCSK9, which is responsible for hypercholesteremia, and then HBV surface antigen, responsible for chronic hepatitis, and also oncogenic CARs, which produce solid tumors. So these are some ongoing clinical targets uh, mm, of siRNA technology. But scientists are not only focusing on liver. So another aspect of this technology is that they are trying to reach as many organs as possible, like as I mentioned, brain, heart, muscle. So to reach there, they invented another process or another way in which they said if we if we bind a ligand to the siRNA, like enzyme bound substrate, so if we add a ligand, tissue specific ligand, to a siRNA, then the ligand will help the siRNA to reach a specific target tissue. So this conjugated system elevate the siRNA technology on another level. So the ligand will help the siRNA to reach a specific tissue. There's a current research ongoing on different aspects, how to treat. Actually, they are trying to treat the neurodegenerative or genetic disorder that are not easily cured by the available drugs. Next. Okay. So the ongoing research helping the scientists to, go on another level to target different aspects like oncology, neurology, and then also the personalized medicine. We all know how much it is important to go for personalized medicine now. The antibiotic will not work in around like 2050. We will die even we use antibiotics. So because of the excessive use of antibiotics, so there are so many types of diseases are coming in our body. It is high time we actually think seriously about personalized medicine. So in that case, siRNA can help tremendously because it is very specific in its acting. So scientists are also trying to do something with siRNA, how to tailor the personalized therapies. Next. Mm, this is one of the most interesting topic I really wanted to discuss. That at your very first class you learned about CRISPR. So what happens if we combine the CRISPR and the siRNA technology? So CRISPR cut a specific gene, but after cutting the gene goes for repair system. It can be a homology directed repair or non-homologous and joining. Two types of repair mechanism. In case of non-homologous, it actually becomes more error prone, as we know. But homology directed produce a very pure DNA. So if we silence the non-homologous adjoining genes by siRNA and then use the CRISPR Cas to cut a specific gene, then the gene will go for homology directed repair instead of non-homologous adjoining. So that we will insert two of them at the same time using a lipid nanoparticle. Then the Cas9 will cut the target gene and siRNA will silence the non-homologous adjoining repair genes. In that case, the genome editing will be more precise. So this is very, like promising side of this siRNA technology. Next. So this is the finishing of my class. I tried to discuss the mechanism and the delivery system and the limitation of this process. So as I said, to use it in full potential, we actually need to the mechanism siRNA follows and the process in which they can disrupt the delivery system and other limitation. Is it not very efficient or does it have any side effects? All of this need to be considered and need to be understood more elaborately to use siRNA in therapeutics. So I have a question for you guys. That next slide, Hafsa, next slide. So like miRNA and siRNA. We talked about miRNA a little bit and then siRNA. So siRNA was discovered in 1998, but Nobel Prize was given in 2006. Whereas mRNA, in case of mRNA, it was discovered earlier, 1993. But The Nobel Prize was given recently, 2024. So why? Like, you can ask yourself or you can look for it, why the siRNA got Nobel Prize in so short time, where mRNA took so long. So this was all about my class. Thank you for listening. I don't know what I can teach, but I tried to share some learnings or some knowledge that I gather from different types of scientific articles. Thank you.
Thank you so much, Ms. Jannatul Ferdous from University of Science and Technology, Chittagong, Bangladesh. And I really appreciate your efforts you put in scientific community as well. And you share your knowledge and even very insightful topic, gene silencing. Yeah, I really appreciate it. And right now, uh, we are moving forward. If anyone have any question, if you note the questions, you can write your questions in chat box. We will answer your question one by one. Thank you so much everyone to join us, and thank you so much for your dedications and passion for learning. And hopefully next classes will be about the bioinformatics and molecular docking. We will provide you complete class with overview, and we will provide the detailed lectures as well for your better understanding, to make that course very beneficial to the student, to the learner, those really want to learn actually. So I hope during all classes you learn a lot of insightful things from international scholars as well. And yeah, I hope, I wish you good luck in your future dinners. Right now, if anyone have any question, you can write your question in your chat box. Yeah, Miss Hafsa, please read out the questions. Can you please?
Okay, sure, sir. Yes, sir, I can. Is I audible?
Yeah, please continue.
Okay, Ms. Janet, here is a question from Rubab Hassan. She's asking, does the dices work only on double stranded RNA? Is this a limitation? I think Ma'am Janet got disconnected. She is not in the class.
Okay, all right. Maybe right now there are some consequences she is facing related to, yeah, network.
Yeah, she is back.
Yeah, can you miss? Yeah, Miss Hafsa, please again repeat. Miss Jannat,
Okay,
please
sir.
ask this question.
Ms. Janet, here is a question that does the dices work only on double stranded RNA? Is this a limitation?
What? If, uh, as, as per my knowledge, I can say that this is the mechanism dicer can detect double stranded RNA. So the question is like, why it cannot detect the single stranded RNA? Like, so siRNA itself is a double stranded RNA. You know, in that case, to be siRNA, it should be double stranded. Otherwise, it will not be siRNA or RNAi technology pathway. This I can guess, or yeah.
Okay.
Murtuza, if you want
Here
to
is a
add something.
okay.
Yeah, yeah. You are exactly right regarding to this question. Kindly read out next question.
Okay.
About
Here is the next question.
personalized medicine.
Yes. Naseeb is asking about how to use siRNA to make personalized medicine better.
Okay, good question. This one. So, what is personalized medicine actually? That it is specific for any specific person. This is the basic concept. Not all human are same, so we need to make different drugs or suggest different medicine for different person. In that case, so you if you have diabetes, you may have mutation in one gene, or you may have over expression of one gene or less expression in one gene. So in that case, we need to produce a siRNA that will downregulate the uh, gene that is causing the disease. So the siRNA will not work for other person that don't have the gene that is causing the disease. So you can say that what about the normal genes? We are not targeting normal genes. We are targeting the diseased or abnormal genes. So in that case, you can use personalized siRNA or for a specific person or a specific genes.
Thank you so much, Ma'am Janal, for answering the question and clearing the queries. Guys, if you have any still question, you can write in the chat box so we can ask Dr. Ma'am Jannat. And if, okay, here is someone asking, can you please briefly tell us about the piRNA?
Okay, so to be honest, piano was not is not so uh, vastly studied like siRNA. So I only focused on the siRNA mainly. Piano also one like a PIWI RNA technology actually. So it use a protein called PIWI. So it varies like same, not more than 30 nucleotide, and it works it comes from the tasposons that work on like viral infection or something like that. I am not so much knowledge in vast area. I know more about sRNA and mRNA
Okay,
more than PRN.
I want to add something to into this. RNA means non-coding region in the genome uh, that cannot generate dicer. All right, PIWI. All right, so that is a non-coding region in the genome.
Then all RNAs are non-coding in that case. sRNA is also non-coding, mRNA is also non-coding. We are talking about non-coding RNAs in the RNAi technology. All of them are non-coding.
Yes, yes. So any question
Actually,
remain?
I'm not seeing any question in the chat till now.
Okay, so we have to conclude our today's class. If anyone have any question, you can still after the class you can write in the comment box, and we will try to answer the question. Yeah, we have next class right now. We have conclude our this class. Thank you so much, everyone. Uh, yeah, Miss Shazad Abdali. Shazad, Mr. Shazad Abdali, we will answer your question. Please write your question in comments after the class. Thank you so much, everyone, to join us and hope for the best.
Yeah, thank you so much, ma'am Jannatul Ferdous.
Thank you so much, Ms. Jannatul Firdaus. I really appreciate your time. We really feel pleasure to have you here in today's class. Yeah.
Allah Hafiz, everyone.
Mr. Mansoor, kindly end the class, please.